we have breast cancer microenvironment model in which we have the breast cancer cells, and we also have immune cell populations. We have stromal cells, so fibroblasts. And we were addressing antibody drug conjugates. These ADCs are very potent and can kill these cells quite quickly. But when we have the 3D model in which we have this tumor microenvironment reconstitution, this changed. So the stromal cells, these fibroblasts, they contribute to the resistance to these ADCs, and we could see that and study that in the 3D model. And this is something you cannot and wouldn't see in a flat culture flask.
Welcome back. In part 1, Catarina Brito introduced us to the power of advanced 3D cell models that mirror human tissue complexity far beyond traditional culture systems. Now we're going deeper into the immune microenvironment, the hidden player that can make or break therapeutic success. From immunotherapy resistance in tumors to unexpected responses to gene therapy vectors, Catarina reveals how understanding innate immunity is reshaping preclinical strategy. Plus, she will share her vision for the future. Let's continue. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Can you give us a concrete example of how modeling the tumor microenvironment in your system has revealed insights about various interactions or mechanisms? How does that work?
There are different types of example. One practical example in a work that we have published in which we have breast cancer microenvironment model in which we have the breast cancer cells, and we also have immune cell populations, and we have stromal cells, so fibroblasts. And we were addressing antibody drug conjugates there. Typically into the models, this type of cells, and this was an anti-HER2 ADC. So HER2 positive breast cancer cells into the cultures, these ADCs are very potent and can kill these cells quite quickly. But for example, when we have the 3D model in which we have this tumor microenvironment reconstitution, this changed. So the stromal cells, these fibroblasts, they contribute to the resistance to these ADCs, and we could see that and study that in the 3D model. And this is something you cannot and wouldn't see in a flat culture flask. Another example, so we also work in neural models, and we are using these neural models to address the initial step of immunogenicity to gene therapy vectors. For example, in this case, the model in which we have all the glial cells, the microglia, which are the resident macrophages, we could identify very transient but intense responses to these viral vectors, so immune response that has been not detected in other models, but that was observed in patients. This is also an example of how better reproducing the human physiology can contribute to identify factors that may be then used to improve these therapies in the future.
Where do you see these models evolve in a few years from now? Because there's a lot happening, I'd say, on the bioprocessing field, how we culture cells, how also on the process, I'd say, analytical technologies, how we maintain these conditions very well. And then on top, we have now to throw in a hype word, AI and machine learning. How will these technological advances affect the way you're conducting your experiments?
I think this type of advanced cell models combined with multi-omics information, right, of layers. Multi-omics information is now also being uncovered, and spatial information. So this is particularly relevant if we think on the three-dimensionality and on the different components of this environment. I think this data will feed AI-based models, and this will give us a more accurate representation of human biology and pathophysiology. We are making essentially observations up to now, and we are comparing with the human, but really to start making predictions, the combination with AI will be very important. Starting by thinking of digital twins of our model itself, benchmarks against patient material, for example, patient data will be important. And then there's a panoply of things that can be done, right, to better tailor treatments according to biological profiles of patients, optimizing dosing strategies, designing drug combinations. I think it will bring a next step into precision medicine and how this precision medicine can be implemented more worldwide.
What is your advice to a smart scientist listening and wondering how could they implement these advanced models? The reason I'm asking this question is we have a lot of amazing tools at our disposal, but it can also be quite overwhelming. And the question is, where do you start? Or how simple can you make these models to still get a benefit out of it?
I think the answer is in focusing on the question one needs to answer. I totally agree with you to what you were suggesting. We should go as simple as possible to answer the question we need. So not so much the focus on the technology as a driving force, but on the question that then leads us to what is the biological endpoint we need to address, if it's the mechanism itself, a potency, a safety assay, and then from then decide on the level of complexity we would require. And I also think that nowadays, with all the tools available, overengineering too early before understanding the cells and understanding the question may be an issue. That's why I defend a system which is as modular as possible, to start simply with tumor, with organ cell-specific spheroids, then to move into stromal immune, but depending on the project demands. And also then to avoid oversimplifying, on the other hand, to avoid treating these as regular 2D cultures, ignoring mass transfer, of course, the importance of the cell sources, and not facing it as a process, which it is, and that needs proper process controls. And this will be important for the reproducibility and for the standardization, which is sometimes what seems daunting with these models.
At iBET, you're collaborating a lot with various pharma companies, and I imagine you also accompany them in certain ways to guide them how to use the models. Can you give us a picture of what kind of collaborations are you doing? How do you run these collaborations? And how do they shape the design and the application of your cell models?
It's true, we collaborate a lot with industry along the years. We were lucky to have several pharma partners that help us really define how we work. Because the truth is that pharma partners bring questions that are shaped by translational needs. How to make the models more predictive, more scalable, more compatible with their development workflows. And these applied questions help us decide a lot model design and to prioritize also the type of interrogation we are doing. It helps us put the models in the real world. And then, I would say, the key to success in collaboration is always alignment and transparency. being very clear on what's the biological question, like I was mentioning, what are the performance metrics, and which are the limitations of the systems we are implemented, and be able to openly share data in both directions is also been very helpful to move some of our platforms from a proof of concept age to really contribute to accelerate translation in those companies.
What kind of companies do you in general work with?
I've been working mainly with pharma companies. And amongst our collaborations, we worked with Boehringer, Ingelheim. We worked with Merck Global Health, which is the social business, German Merck. We worked with AbbVie in the United States, with big companies that are developing advanced therapeutic and new therapies.
Do you also work with smaller companies or startups?
Yes, we do. These collaborations I mentioned were very long-term collaborations along several years and developing from scratch models that were not available to answer questions that were not addressable by existing models. We've also worked with small companies over the years, typically on more specific questions they have on their compounds with models we already have implemented.
Now I have a question about if there are any pitfalls to avoid, because as we have alluded to before, we have a lot of technologies, but where do you start? What do you leverage? What is hype? What makes really a difference? Can you point out 2-3 pitfalls we should watch out for?
As I was mentioning, I think over-engineering to technological driven before understanding the cellular system and what you need cells to be doing and which cells. I think the cell source is also a very important aspect because depending on your question, you may not get from the cells you're using never your answer for how much you engineered them for. That's also an aspect. As a third one, I would say not validating enough, not characterizing enough, moving immediately to the application without having your system properly characterized. I think it's important.
Before we wrap up, Catarina, what burning question haven't I asked that you are eager to share with our biotech community?
I think we were quite thorough. It was a good conversation. And you can see how passionate I am probably about the topic, right?
Yes, absolutely.
And it gives context also to my work because being integrated, my appointment is with Nova University in Lisbon, but my lab is also affiliated to iBET, and it's through iBET that I have the collaborations with industry because iBET is a partnering research organization that bridges engineering and biology to accelerate biopharmaceutical development. So this is an ecosystem in which we combine expertise that goes from cell line development, cell bioprocessing, and advanced analyticals, and then closing in our translational models under the same roof. So this allows us to have access and to have a collaborative environment in which it's easy also for us to work with the advanced therapeutics from the antibodies to the gene and cell therapies. That's it.
This has been great, Catarina. What is the most important takeaway you want our listeners to walk away with from our conversation?
I think the takeaway is that the advanced cell models are here. They are here to stay. And I think they are here to help shape translational research in the future and doing, in the meanwhile, some transformation in the drug discovery industry.
This was great, Catarina. Thank you so much for letting us into the world of 3D cell models. And where can people get a hold of you?
So I'm in LinkedIn, right, as much everybody, through iBET's website, ITQB NOVA's website, but I would say through LinkedIn, it's the best way of contacting.
Excellent, Smart Biotech Scientist. You find the links in the show note. Please use this opportunity, reach out to Catarina. And Catarina, once again, thank you so much for being on the show today.
Thank you, David. It's been a pleasure. Bye-bye.
That wraps up our conversation with Catarina Brito. Her work exemplifies the future of preclinical research where complexity becomes predictability and where patient-specific insights move from aspiration to reality. If this episode sparked new ideas for your own development programs, I'd love to hear about it. And please, Take a moment to leave a review on Apple Podcasts or wherever you listen. Your support helps us bring more innovators like Catarina to the biotech community. Thank you so much for tuning in today. Until next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.